Good morning, everybody. Welcome to ATP 101 with OSP. My name is Virginia Warnstaff. I'm the CTO here at OSP.
And I'm Mark Demeter. I'm the Service Manager and Applications Microbiologist.
So today we are going to be doing a live hands-on demo of our ATP testing technology. Just to let you guys know, we don't have a huge production budget, so this is going to be very similar quality to a YouTube or one of your video conferencing. Because Mark and I are going to work so closely together today, we are wearing face shields rather than the surgical masks. So we have a barrier in alignment with our COVID-19 safety procedures, but that you can still see us and hear us throughout the webinar. So before we begin, I do want to give a little introduction to OSP. I noticed that there's some people who are new to OSP. We've been around for 30 years. We were built on the research and development of our flagship product, TK7, which is a biocide. And over the years, we've expanded into different markets and added different product lines to our business. But in the recent years, we have added a microbial evaluation services team, which we call our LifeCheck team. Mark heads up that team, and in our lab here, we offer testing like Atp testing as a service or as kits. We have in-house DNA testing, Qpcr, and 16S sequencing. We can do kill studies, we do program evaluations, we can do reviews of field programs, you name it. And very shortly, we're also going to be expanding and offering COVID-19 testing for environmental samples. So the reason why we started this service group is not only to support microbial mitigation or inhibition, but it's also to further development and understanding of microbes in our industries. So I'm going to start by handing it over to Mark. Mark's going to give us a little overview of ATP specifically and where it fits into the broad world of testing available to us in our industries. As we go along, I do want to note, please, by all means, ask questions as we go. We're going to stop at certain places to take questions. Moira is our Marketing and Communications Director. She's going to be moderating today, so you may hear her voice in the background. But Mark, take it away.
Thanks, Virginia. One quick thing on the questions. I believe we have some prizes if we have any outstanding questions, do we not?
Absolutely. This is a wonderful OSP decorated sock. And as questions come in, we may use this to get a few more. And this will be sent out to whoever answers first with certain questions.
Well done. All right, so yeah, let's get started. The topic of today's webinar is ATP testing. But what is ATP? What does it stand for? Well, ATP is an acronym for adenosine triphosphate. And adenosine triphosphate is a biomolecule found in all living cells. I like to think of it as the energy currency of life. You use ATP to conduct activities within your cellular processes. So every single organism uses ATP, whether that's microbial, like a bacteria, or even mine and Virginia's cells. We all make use of ATP. Virginia alluded to this a little bit earlier, but we actually have a full suite of testing technologies here at OSP, and they kind of are complementary and fit a different niche in different ways. And that's really because the information yield that each different testing technology gives can change. So of the 4 testing technologies that we offer, we have our LifeCheck media, which is also bug bottles, culture media based type of testing. And that is going to give you the lowest information yield compared to the rest. The biggest reason for that is because as a field of microbiology, we can only actually grow approximately 1% of the microbial members that are out there in the environment. So you miss out on a significant portion of what you should be testing for. Moving on up that information yield scale, we find our ATP testing technology, again, the focus of what we have to discuss today. And the important thing to note about ATP testing is that because a biomolecule is found in all living cells, it is going to be far more, the resolution of that it's going to capture is going to be far greater than the culture media. However, it doesn't tell you what type of organism is present. So you're going to get a microbial number of an active microbial load, as in a living representation of the microbes in your sample, but it's not going to tell you who they are and what they're capable of doing. So that's where some of our DNA testing offerings come into play, the Qpcr and 16S sequencing. That Qpcr method, that is going to allow you to enumerate and target microbial subgroups of interest. So for example, if you're interested in the souring potential of your microbial community or a corrosion standpoint, you might be looking at sulfate reducing bacteria and counting them using a DNA marker. Additionally, we have other targets, ranging anything from methanogens to iron-related bacteria. Lastly, we have the 16S sequencing testing technology. I like to think of that as our flagship testing technology. It's going to give you the most information yield as compared to any of these other ones. And it's really meant for that higher level deep dive or troubleshooting type experience where you want a complete understanding of all the microbial names within the sample, so that skilled microbiologists can actually go into that list and start divulging the functions and activities that those microbes might be doing in the community.
And knowing the limitations of what you're getting with the different information yields for this testing helps you choose which one is the right one for your application. It doesn't mean that there's a silver bullet and one supersedes the rest. It's just simply understanding it so that you can get the best data for the decision that you need. So let's go into the ATP testing. We are going to be walking through our procedures. We have an ATP handbook that some people may be familiar with. This does come with some of the components of our kit. We're going to walk through from beginning to end the water testing, the planktonic, meaning we're going to target free-floating microbes. There is 2 versions of this book, so there's going to be some numbering differences if anybody's following along. But fundamentally, the core of what this testing is going to be the same for any of the applications that we use it for. And we're going to give some context and talk around the different changes or nuances, depending on the type of sample or the procedure you're going to use to extract it.
Yeah, absolutely. We'll try to give you some tips and tricks that we use ourselves, that some of our clients have shared with us in the past for dealing with difficult samples or sample matrices that you might not even think are capable of being tested with ATP. But before we can get started with a live demonstration, we need to be cognizant of our ATP. Thanks to COVID, Virginia and I are already wearing our wonderful masks or eye protection. However, we are going to put on nitrile gloves. So in this case, this is actually serves dual purposes. One, you're protecting yourself from your sample. That can be quite important if you do have harmful chemicals in your samples or not. But anytime you're doing microbial testing, you also need to be cognizant of the fact that there are microbes on your skin and your saliva and so on that can be unintended sources of contamination into your samples. So you want to prevent that as well. So we wear our gloves and our face shields in this case to protect ourselves just as much as we want to protect the integrity of our samples.
So what are we testing today, Mark?
Well, we're going to start off with that planktonic test matrix. So we have a liquid sample here, labeled sample. And we are going to enumerate the active and living microbial load in this sample using our planktonic ATP kit.
Okay. So when you order these kits, you'll come with all of the disposables, the plastics, the reagents. So everything that we're going through here today, we'll do our best to highlight the name of it. I'm not sure if you'll see very much of it with the camera angle, but you can correlate it right back to the kits based on which one you are purchasing. All right.
All right, so I've provided Virginia here with a 50 ml syringe and a syringe filter. And the premise of what she's going to do here in this first step is, well, syringe filter our liquid sample so that we can collect microbial cells on the surface of the filter membrane.
So you can take your syringe and dip it into the sample and pull the sample up. Depending on how big of a sample or the mouth of the container that you're using, that might be difficult. What I prefer to do is to pull the plunger out. Never pull air through this filter, so always make sure the plunger's out when you're changing reagents or putting it on, like so. Have your waste container. I place it on my finger and hold it like this. Mark, would you mind opening my sample?
Sure.
Make sure that it's been shaken up as Mark did, and then we pour it in. The more you can filter, the better. So we're going to start and aim for 50 mils. Right to that line. I'm going to place my plunger back in, holding it over the waste receptacle. Maybe there's a little bit of a pop. There it is. All right. And then simply push the sample through. You can see the fluid spreading out across the top of the filter. That's nice. You don't want to push too fast or too hard. If you are using your own disposables, make sure you have a lower lock on the end of your syringe to make sure that filter's on there tight, because we have had experiences where it'll pop off and make a mess.
How are you doing there, Virginia? Are you going to get the full 50 mils?
I don't think it's going to work.
Yeah, there is some sediment in this sample, which does make it a little bit more difficult to filter syringe. If you've been skipping the gym because of COVID, just like Virginia has here, We do have one tip and trick that can work quite well if you use a little mechanical leverage. So as goofy as this may look, you can actually put that syringe inside of a caulking gun, and as you start activating it, it'll actually squeeze that syringe for you in short time.
We do use that in our lab annotation, and a lot of times too, it can come in very handy if you're doing a lot of samples in a day, because it can get a little bit strenuous. Hands might hurt. But I'm going to say that I'm at the place where I can't push any more through, Mark.
All right, what'd you get?
20 mil.
20 mils?
Yeah.
All right, the first thing we should do here is now record the volume that we did syringe. You want me to hold that for you?
Sure.
Because the answer at the end of our test here is going to be in units of a number of microbes per unit volume, we do want to record the volume that we have sampled from, so that in our calculation we take that into account.
Now you note that I put 50 mils into the syringe, but I'm only recording the 20, because that is only the amount that we got through.
Right. And the other thing I should add is that all these fields that Virginia is filling out on that whiteboard, all of those fields would actually be present in our LifeCheck ATP calculator tool that we do provide with purchase of our reagents.
So now the important part of this is to keep this filter. So because I push so hard, I do back it off just slightly so that when I take the filter off, I'm not going to get splashed. Again, hold it over my waste receptacle so I can pull that off. Ooh, success. Now you can either try to push the rest of it through if it's not too much blocked, or you're going to want to take the plunger out so that you can empty the syringe, because we're going to continue using it while we push different reagents through that filter. All right. Again, don't put the filter back on until we pull this plunger out. Place the filter right back on. And we're ready for the next step.
All right. So at this point in time, we have our microbial cells collected on that filter membrane. And the next step we're going to conduct is a wash step or a rinse step. So the rinse reagent is an additional add-on that you can purchase for our kits. It is an add-on that I highly recommend you use on all sample points. The intention here is that we're going to be providing an excess amount of our rinse reagent and passing it through that filter syringe to free our trapped microbes of any other chemicals or contaminants, residual hydrocarbons, for example, that might become inhibitory to the downstream process of actually measuring the amount of ATP.
Basically, we have really nasty samples that we tend to work with in our various industries, and we don't want any of those contaminants to block that reagent, that lysing agent that cuts these cells open, from accessing all those cells. So we're just trying to get rid of anything that might block the next reagents from doing their job.
So I'm using a bald pasture pipette here to transfer some of the rinse reagent to the syringe there. This isn't an exact measured amount. It should be a couple mils or an approximation. Again, the idea here is that we're just passing this through to clean it up. So it doesn't have to be an exact measure from this guy.
So again, pop the plunger back in, push it through. I can see that my first drops are a little bit hazy. And hopefully as we continue pushing this through, they will start clearing out. Now I am putting a lot of pressure on this because that last sample was particularly nasty. We did want to make sure that we use representative samples that we may see in the field as a worst case, so you can see some of the challenges. So I'm just holding pressure on this and allowing it to drip through. Again, every drop is getting a little bit clearer, which means I am cleaning it up, and that's what's important. All right.
All right, so now that we've cleaned those microbial cells captured on this filter membrane, the next step in the process is to crack open those microbial cells, releasing all of their cell innards, including those ATP biomolecules that we actually want to count and measure. So to do that cracking open of the cells, we're going to use our reagent X. And now this is a lysis agent, and we do want to add a specific volume of this to our filter syringe apparatus here so that we can take that into account in our calculations. And to do that, Virginia is going to use the brown 1 mil fixed pipetter provided in our kits.
May I have my field container as well?
Absolutely.
So in this step, we don't want to put it to waste. This is where we're actually capturing that ATP. So there's two places where you can filter this last step into. The kits provide field containers. This is what I'm going to filter into. This is to allow us to stop at this step. And you can gather all of these within the field, put them into your cooler in your truck, and then take it back to the lab where we use the luminometer, or to a facility to take the reading.
Right. So if you have a luminometer on hand with you, best practice would indicate that you would finish your testing at this point and actually take the reading with your luminometer, which means that you might extract your ATP using Reagent X directly into this tube here, the Reagent D tube. In Virginia's case, we're going to move forward as though her only luminometer, the only one she has access to, is back at a field office somewhere, and she's out in the field. So she's going to elute into the field container. Notice her pipetter technique. She didn't hold it sideways or dump it upside down, and using a fresh tip each time.
All right, this one you want to push all of that fluid and the air through to the end. So you're going to see often some foaming that comes through. This is a tough one. Look at that. And that's okay. Push this plunger all the way to the bottom if you can get it through. Make sure you get every last drop. And now you're finished with this. And we have our sample captured in the field container. And this can stay in cool conditions for not freezing for 3 days, maybe up to a week. If you're doing a lot of sampling, a lot of gathering, and you need to get it back to a central location. And then once we get there, we're going to take the reading. So what we've covered here is the extraction steps. We're going to take a pause, see if there's any questions, and then we'll move on to reading our sample.
So we have two questions. One was in regards to Mark's caulking gun, and a question in regards to at what point do you get concerned about overstressing the syringe and having it break apart?
Yeah, so these are all Luer lock syringes, and the syringes that we provide have about 3 threads on them. So they will actually take a fair amount of force to crack open and break. I have had it happen a few times, so you definitely don't want to go too far. And we do actually have a methodology to help you deal with samples that are so viscous that filtration just really wouldn't be the best way to go. So we will go into that as soon as we finish with this demonstration here, and we'll talk about that option. But to the person that asked that question, that's a very valid point. So I don't advocate that you push so hard on your caulking gun that you blow anything up. It certainly makes a mess. It's really more in that situation where you're doing maybe a dozen different samples that are a little bit more viscous, and frankly, it just gets tiresome to try to filter syringe that without some mechanical advantage.
Okay, another question, and this may be answered further along, but I'll just bring it forward, is with regards to recycled water facilities, what do you recommend in terms of testing?
I guess that depends on exactly what you are testing for. Again, microbial load is an important part of understanding what your microbial inhibition or mitigation programs might look like. So this is actually a very easy test for anything that's planktonic and water that is treated to push through. But if we can provide some more context on that, we can engage it absolutely and provide more guidance.
And I'm just going to do one more here and then we'll carry on. And don't worry, for those who put in questions, they will get covered as we go through. But the question here is, with regards to the reagent, do you not have to use 60 milliliters of air after the rinse reagent?
Yes, I would say push through as much as you can. The air is absolutely push the plunger to the bottom as you would in the next step, like I mentioned. If you can't, it's not going to be detrimental, let's say. So there was a lot of sediment in that sample, as you saw. I pushed through as much as I could, and then we move on to the next step. Go from there.
Okay, we'll continue on with questions later on, but I'll let you guys carry on, and then I'll bring forward questions at our next break.
Thanks for the questions.
All right, so we're going to talk a little bit about the technology and the instrumentation first before we dive right back into our live demonstration. I have in front of us here a luminometer. So this is effectively a box with a spot for these little cuvettes or assay tubes to go into, and a little handheld unit that controls it and reports back the data that comes from it. So that's a good point here. The data that comes from this is relative light units, or RLUs. So the reaction that we're going to take advantage of today in order to determine how much ATP is in our sample unknown is a luciferase reaction. So what I mean by that is that there is an enzyme or living component, that's our reagent Z. This is a luciferase enzyme. And when this meets up with ATP, it actually produces light. So this box works in kind of like a reverse flashlight mode, where instead of giving off light, it reads the amount of light that the reaction between the luciferase enzyme and the ATP happens. If you consider that the more light that is produced means that there's more ATP. More ATP would mean that there is a higher microbial load in your sample being tested. That's where we're able to backtrack from an RLU value all the way up to a number of microbes per unit volume. To take that measurement and to do so in a way that has high repeatability and assurances that you've got quality data, we have to run a couple controls in our luminometer before we get started. So we're going to run two controls. One is going to be a blank, ensuring that the instrument itself isn't contaminated, doesn't have a high level of background reading that would hurt our lower levels of detection. And then we're also going to be running a positive control to indicate to us that our enzyme, which again is a living component, is healthy and happy. If these enzymes are left out in the heat and say the dash of your truck, what happens, Virginia?
They degrade very quickly.
Right. So we want to make sure that this guy is happy and healthy and ready to go for testing.
So these enzymes are very much like what you find in fireflies. So it's a synthetic version of that. And for anybody that is in our Texas crowd here, again, that heat and working out in the field, that will degrade it very quickly, like within hours. So that's part of the reason why we have that first step, so that you can stop and take it to a facility that has temperature control. Otherwise, I would suggest between every use, toss it into a cooler or an air-conditioned environment.
Right. But prior to that, as in prior to having the ready-to-go enzyme in a bottle, it's actually shipped to you in what's called a lyophilized or freeze-dried form. So I have here 2 vials. This is how you would see your reagent Z on delivery. And we have the lyophilized version here. It's a white powder inside of an amber bottle. And this could be stored at room temperature, experience different temperatures as a whole, and be okay. So this guy, you don't have to worry about the heat, at least until you've added the buffer, literally pouring this into this one, activating your enzyme. And at that point, you need to be worried about its health.
You can keep that enzyme in the fridge in between doing testing rounds, and it'll stay for quite a long time.
All right, so I think we're ready to get on to our standards testing. So the first thing we're going to do is turn on our luminometer. That's the yellow button on the left. It'll take a few minutes to turn on. In the meantime, I'm going to place a blank or empty assay tube into our instrument. Once it turns on there, Virginia, if you want to hit the green button to the point where it takes a reading.
That was 3 pushes. And then it's going to count down for 10 seconds.
And we'll record this value as it comes out of the instrument. In our spreadsheet, this is referred to as the BRLU or background RLU.
I'll let you hang on to that.
All right. And we have a BRLU value of 5. So this is exceptionally low. This is where we would like to see a luminometer come up for a background reading in one of our assay tubes. If this was significantly higher, like on the order of maybe 30 to 40 or even into the hundreds, there could be a couple of different things that went wrong here, and you should stop at this point and correct or mitigate some of these things. The first is that your luminometer might be dirty. If you are in a habit of pipetting directly over top of the luminometer, sometimes things can splash and drop inside of it. If that is the case, we do offer a cleaning kit for these luminometers, or you are more than welcome to ship them in to us here at OSP, and we will clean them for you and check their calibration at the same time.
The other thing that might happen if you get a very high reading on this is that these are pretty robust, but if they have been banging around or being squished within different equipment and things like that, and they start getting scratched up, they will start giving a higher reading. I'd say it's around 30 RLU that you might toss it and grab another one.
Yeah.
That's probably the max that you want these to read at. And it is simply just grab a new one and start over with the next.
All right, so we are happy with our background reading of 5 RLUs. And the next step here is going to be our positive control. So I'm going to take my assay tube and just place it back into the corner of my rack here. Again, best practice isn't to pipette directly into the machine. And Virginia is going to help me out first by pipetting 100 microliters of the enzyme, the reagent Z, into this assay tube. Once more, we have a preset micropipetter that is going to move that 100 microliters of fluid from this bottle into our assay tube. And now we need some synthetic ATP in order to react with that luciferase to produce light that we can pick up in our instrument to ensure that it's working. So you're provided with a little eyedropper of synthetic ATP and 2 drops. Hope I got 2 there. I had a lot of coffee this morning, so that was a little rough. 2 drops is going to be 100 microliters. Virginia gave it a little tap, a little swirl to mix it up, and dropped it into the luminometer. I'm going to hit the green button once to take a reading. And now we're aiming for 5,000 RLUs here as an indicator of a healthy enzyme. But to be honest, these things are typically around 15,000 microliters or RLUs or higher. And for this one, we managed 21,184. So very healthy enzyme, ready to go.
Mark, what happens if it's below 5,000?
That likely means that your enzyme as a whole has started to denature or is just no longer effective at converting ATP into light or producing a light signal in its reaction. So at that point, it's garbage, unfortunately, and you need to start over with a new or fresh thing of enzyme. All right, so we've done our 2 controls. We've got our blank that tells us that our instrument is ready to go and doesn't have a high level of background noise. We've done our positive control that tells us our enzyme is healthy and ready for testing. Now we're ready to go ahead and figure out the amount of ATP we actually have in our unknown sample.
Excellent. So, like I said, this could be sitting for a little while, waiting for you to get back to a certain spot. And if you haven't pushed directly into the tube, at this point, we're just going to take this tube of our reagent D. We're going to pour it into the field container where our sample's collected. Place that lid back on and give it a little swirl, make sure it's good and mixed. It's easier to pour that tube into the field container than to go the other direction. Suggest that you sample it as you go along, make sure you don't lose count of who's who in these little containers. And then we can start taking our reading.
Yep. So at this point, Virginia can add 100 microliters of the reagent Z to a fresh assay tube, making sure that she uses a new pipette tip, doesn't cross-contaminate in any one sample from another. And after she's pipetted 100 microliters of enzyme, she will pipette 100 microliters of the sample that she just added the reagent D to.
Again, give it a little bit of a swirl.
Pop it in. I'm going to hit the green button.
And this is going to be our final reading.
All right, 163,494 RLUs. All right, awesome. We have a reading now of the amount of ATP in our sample. 163,000 RLUs. What does that mean? I don't know. So the direct value that comes out of the instrument isn't what's useful. What's useful now is taking that information and calculating it back to a unit that does provide information that you can then make a decision on, understand the mitigation processes you have in place and how they're affecting the microbial community. So the calculator spreadsheet that I mentioned earlier will automatically do this. Because Virginia is super at math, she's just going to do it all in her head right here. So 163,000 RLUs with all this other background information and standards taken into account will give you a mass of ATP of 3,859 picograms. Is that right, Virginia?
I think so. Carries a 3, yeah.
Yeah, magic of television, am I right? So we're getting there. That is still quite useful. And there are folks out there that do actually trend their data as a mass of ATP, a picogram of ATP per mil. And there's absolutely nothing wrong with that. You can certainly make decisions and get a trend again if you've got an increase in mass of ATP or a loss. That is an indicator that you do have a flux in the microbial load within your sample. I do, however, like to take this one step further and convert it to a microbial equivalence, or a number of microbes per unit volume. Because at the end of the day, that's really the heart of what we're after here, is the number of active and living microbes in your sample. So once more, that calculator tool will automatically do this calculation for you.
What's the log number on this?
10 to the 6.
Okay.
All right, so that's actually a very important thing to distinguish here, is that what we're really concerned about isn't so much the number in front of the scientific notation or of the exponent, but the exponent itself. As in, we have 3.9 million cells in this sample per unit volume of 1 mil. To say that I have 3 million or 6 million, yeah, 6 million is twice as much as 3, but that's really not the take-home point. The take-home point is that I have millions of bugs in my sample. In the same way that if you were to conduct culture media testing, like bug bottles, this would be the same result as having 6 bottle turns. It's not the number of microbes necessarily in front of that exponent that matters, but again, the number of bottle turns. You might also think of this unit as a CFU per mil, or colony forming units per mil. And the last thing I just want to say about the concept of the microbial equivalents per mil is that you can actually use this across all of our different testing technologies. So whether you're doing culture media, ATP, or Qpcr, you can be given results in that same unit. So it is a universal unit to that point and allows for some comparison, of course, with caveats that tests measure different things, but at least the units are consistent.
It allows us a common language no matter which testing technology that we're using. The other thing that we utilize those results for, and it can be in the picograms per mil or in the log or microbial equivalent format, is for contamination levels. So we work a lot in the oil and gas sector. We have generally accepted contamination levels that we use, low, moderate, and a high. Again, I may get some discussion on this, but typically around the log 5 is a moderate level, below a log 5 is low, and above a log 5 is high. Now, that is not an indication that you are or are not going to have a problem based on those contamination levels. It's simply a general guideline that has been put together through experience and work in this industry to give you a baseline to start from for the understanding of the risks in your system. So again, if you start there, there may be some systems that are really harsh, high temperatures, not a lot of nutrients. You can actually have a really high amount of microbes and it not correlate to any direct issues in your system. On the other side, you may have a facility that is full of nutrients, a great temperature for microbial growth, and you may have to move those limits down lower because even a low amount of microbes could easily flourish and become an issue. So again, use the contamination levels as a baseline or a place to start, and then adapt them to your specific needs and your specific applications. And trending is a really good way to do that.
Right. So that's the Planktonic testing methodology in a nutshell. I did allude earlier in this webinar that we do have some other methodologies or adaptations that allow you to deal with other sample types. So Virginia and I will go in and right now and talk about some of those. We're not going to do full or complete demonstrations of each of them, particularly at the measurement of the ATP steps, because once you get to the extracted ATP point, it's all the same in your interactions with the actual luminometer. So one of the first ones I want to talk about, and we kind of brought this up earlier, is that what if you couldn't squeeze or filter syringe 1 or even 5 mils of your sample? When you're starting to sample such a low volume as that with this methodology, the Planktonic one, you might start to wonder if you're missing microbes or if you're going to be too close to your limits of detection. Or maybe your sample is just so viscous, like the sample we have for you here today, Virginia, that you're not going to be able to syringe filter any of that. So for that, we do have what we call our Sessile soak method. And this is a workaround that actually makes use of our Sessile ATP testing kit.
So you saw me struggle with that first sample, but we still got through 20 mils.
Yeah, and that was enough.
Yeah, and if you can get through some, again, just make sure you're using these consistent procedures. If you're going to use the filter procedure for one of your tests in a group of testing or ones that you're comparing, stick with it. Otherwise, switch and do them all with the other procedure.
Right. So in the sessile soak method, there is no filtration step, of course, because that's what we're trying to avoid. So we go straight to the lysis step. And for that, we provide 50 ml Falcon tubes with 10 ml volumes of our lysis agent or reagent X. And what Virginia is going to do in this case is carefully pour some of her viscous sample into this tube.
I don't know if you guys can see, but it is quite viscous. All right. And I'll put in a few more. 5 ml.
5 ml, perfect. So I wouldn't recommend going over 10 ml of your sample into this tube unless you add more reagent X, which remember, you do get reagent X in a larger bottle, so you are certainly within your means to do that. But really what I'm getting at is you don't want to over dilute your reagent X in the sample and then really harm its ability to lyse open cells. So at this point, now that I've introduced my sample to my reagent X, I'm just going to kind of gently rock it around. If this was a sample with a lot of oil organics, you'd see that oil starting to coat all of the plastic. And the concept here is really just trying to have your reagent X contact as much of your sample as possible. You're going to do this for 10 minutes.
And that's it. And then you go into the next step, which you take your sample out of that and go into your interpretation or your results.
Yeah, so it'll actually be 1 ml using your ground pipetter into your reagent D, and then from there, your 100 microliters with your enzyme into that guy there.
The other thing to note is that we typically use this procedure when you have really viscous fluids, and there's viscosity there because perhaps it's a sales oil, or maybe it's a gelled frac system. Microbes only live in water, so they're only going to be in the water component of that total fluid. So if you have part of that fluid volume that is not water, you may want to provide a water cut at some point in your calculation process to ensure that your microbes are only associated with the amount of water. And the reason why that is important is if you're looking at microbial levels over time, you may see a rise in microbial levels, but it may just be that your water cut is getting higher. So having some knowledge of the types of fluids you're using. Also, in a lot of the hydrocarbon type fluids that we're testing, you can spin it and pull the water cut off the bottom. Is there any other things that you use?
No, the water drip on the bottom, maybe trying to get a longer pipetter if you have one down through the floating oil suspension. The only other comment I do want to make, though, is that anytime you start diverging to some of these different methodologies, like the modified soak that you just saw here, it's important that if you're going to be comparing or cross-comparing different samples, that you stick with the same methodology. There's also a separate tab on the calculator spreadsheet for this test. So just make sure that you're doing everything or treating all of your batch of samples in the same manner.
And as we get into the next step as well, just know that all of the calculations that we use are fully available in the handbook, whether you use it digitally or have your own version. And when you go into the worksheet, we do leave it open in certain places so that if there are amendments made to the procedure with the guidelines there, that you can make sure that your calculations are correct. And if you have anything that's outside of the procedures as we go through them, please give us a call. We would be more than happy to try to find a way that this can work for you depending on your samples.
Right. So that's the sessile soak method. And I did mention that that actually is an adaptation from our sessile or solid testing kit. So now seems like a good time to actually tell you about our solid testing offering, which of course is the larger Reagent X tube with the 10 mils. And the intention of this guy is so that you can test a variety of different solids, whether they are pipeline scrapings or pig returns, debris, that kind of thing. I can measure a mass of this and actually pour it into my Reagent X tube. Same thing, give it a shake, allow it to sit in there for 10 minutes, and any surface attached microbes on those pipeline scrapings are going to lyse and then release their ATP for downstream measurement. The other place this is commonly used is in coupon assessments. So imagine, if you will, this bolt is my coupon. Obviously, I want to be manhandling it quite like this, but the idea is that you would simply place your coupon with your biofilm on it, or your sessile microbial community, into that tube, rock it around for 10 minutes, exposing all of that nice sessile microbial community to the lysis agent, and once more conducting your ATP test using the luminometer at this point. We do have one other application for that sessile testing kit, and that is swabbing. So I have here a pipeline cutout. And in the position just below the letter B there is a fairly aggressive pit. If memory serves right, this pit was about a 70% wall loss that occurred over just a few months. So there was some aggressive corrosion happening here. And the client question was, of course, could microbiology be a part of that? And from a microbiologically influenced corrosion perspective, the presence of microbes alone, which is what ATP testing is going to test for, is not by any means sole evidence that MIC is occurring, but it is at least a first step into understanding if there's even microbes present within that pit as compared to the parent surface. And then, of course, we can complement that with our DNA testing technology to see if the types of microbes that are able to influence MIC are there. But backing up a few steps, to conduct ATP testing on the surface, you can get a surface testing kit from us that comes with swabs, sterile swabs, and a soak reagent. So this agent is used to wet the swab. So a sterile swab that I'm taking out of the tube. Be careful not to touch any other parts. And this is only going to be immersed in this soak reagent one time, because I don't want to cross-contaminate it. And then I'm simply going to swab the surface area of my pit. So in this instance, because I haven't measured the surface area that I am swabbing, I am simply going to report my answer in the units of cells per pit. That might not be as useful to you as actually having a surface area. So if you don't know the surface area that you are swabbing, one of the things we do provide are these handy little cards. They can be at least ethanol sterilized, and they are 1 inch squared. So that allows you to swab an area and know the surface area that you are testing.
Now we know that when we have a pipeline failure, these get cut out, preserved, and sent in for metallurgical testing pretty quickly. A lot of that will remove that microbial community. So the faster that you can perform your swab test, meaning in the field if possible, the more accurate those results are going to be.
Right. So now that I have my swab here completed, I'm going to take one of my Reagent X tubes, place the swab in it, and just carefully snap it off in such a manner that I can replace the lid and allow this lysis agent to cause its effect and release ATP from cells over that 10-minute period.
All right, so that covers all of our different procedures for the different applications. We're going to pause one more time and see if we can answer some more questions.
Okay, so we have a series of questions. I'm just going to, I'll go through 5 right now, and then we'll cover off the rest at the end. But first question is, with regards to deceased cells that still have ATP present, is there any way to discern between actual viable cells and those that have recently died but not lysed?
Yeah, so this actually brings up a larger conversation as a whole, which I don't want to get too far into the nitty-gritty on this today. But there's a couple of comments I want to make on this. And the first is that ATP is an expensive biomolecule to make. So a highly inactive, dormant, or dead cell isn't going to be making a lot of ATP in the first place. Once a cell becomes dead, it will eventually lyse and destroy, at which point it will release its ATP from the cellular content, the cytosol. And at that point, it's free floating. If you're doing the Planktonic test, you're actually going to be filter syringing and removing all of that free floating ATP and only actually measuring the cell bound or interior ATP, which means you're no longer accidentally capturing those dead or dormant microbes that might have some trace levels of ATP left over. So that kind of resolves that concern. And then the only other thing I would say about the viability piece is that viability in a microbial cell is a tricky matter because of things like dormancy, persister cells. There's a whole theory out there about being viable but not culturable, in that even if you provide a dormant cell with the right nutrients and growth conditions, they might not necessarily pop back, but in different tests, whether it be staining or ATP, they might not look as alive. So the important take home here, though, is that these steps do prevent unwanted detection of free floating or extracellular ATP and only go after ATP from active and living cells.
Okay, so the next question is, in the field, can you not dispense directly to the tube?
Absolutely. We simply provided the field containers. It might be a little bit easier depending on the types of environments you are working in the field. If you have that dexterity and you can go right in the tube, I encourage it, absolutely.
Next question. We used to measure ATP in cooling water monitoring to distinguish between living cells and ATP from dead cells. What is your opinion on this?
Use ATP to distinguish living from dead.
So I think this is in reference to a different form of ATP testing, perhaps the hygiene method, where you take 2 ATP readings and then your final is what is the difference between the two. The difference between that style of ATP testing and this one is that we account for all of those differences within this entire process. We also have that positive and negative control that we put in here so that you're getting a more accurate reading on that actual picograms of ATP, where some of those simply report in an RLU.
Right, and that filtration step again in the Planktonic one, which we'd be using in this water scenario, would remove that extracellular ATP from the dead cells.
Yeah.
Okay, next question. Do all bacteria contain the same amount of ATP?
No, ATP loads are going to be highly dependent on the type of bacteria that's present, where they are in their growth cycle, and frankly, just how active their microbial or their metabolisms are at that point.
The conversion factor that we use for the microbial equivalents, maybe this is where the question is going, is based on the ATP in an E. coli grown in a lab. We've done a bunch of background work on looking at what those differences are among the microbes in our industry. It's not significant enough that this is going to give you a reading that's not going to be accurate enough to make decisions on.
Yeah, especially at that log level, I will say that. I've done some of these experiments with pure E. coli cultures. And again, the amount of ATP that you do get from cultures in different cycles of the growth phase will change. However, that tends not to be large enough of a difference to change the log extent to which you're reporting.
Yeah, and that's another reason why we use that log reporting. It gets rid of some of the noise of this as well.
Okay, and last question for just this section, and then don't worry, we'll get back to the rest of these, is how often do you run background checks on your enzyme? Every hour during the day, or what's kind of the general timeline, if you will?
Yeah, so you don't have to run that standard for every single sample within a batch of samples. So let's say I collect 10 samples and I want to measure them all in this instrument. I'm going to start off with one background and one standard. Running the background on each can be important to test the tube that you're using for each individual each individual sample. But the standard itself to test for that enzyme health, that's really a batch effect thing, right? So if I'm doing 10 samples and it takes me half an hour to do them all, I can be rest assured that in that time my enzyme should still be healthy. If I come back with another batch of 10 samples 2 hours later, you bet I'm doing another standard RLU test to make sure that enzyme is still healthy.
And part of the reason we let you know that temperature does have effect on this is to, take a look around in your surroundings as you're doing this. If this is something that you are doing in a really hot summer's day, and you're worried because, oops, I forgot to stick it in the cooler, I left it on the tailgate, take another reading and just be assured.
Yeah.
All right. So that goes through all of our procedures. It goes through our kits. I think we've covered most of the DNA side of the technology. If you have any questions about that, Mark and I are always available for any questions as well. Give us a call, send us an email. We also have classes on these topics that we can go through and talk more about some of these questions, like Microbes 101 or sampling and handling. We can get into reports and interpretation and how you report on these things. We're always available, and we love it. We would love any feedback. We're going to get into a few more questions, but we did want to let you know that your feedback is really important. Mark and I want to know if we're worthy of our own YouTube channel or not. Please let us know.
I'm trying to get my own trailer, so.
Again, if there's more questions, we're going to be sending out a few prizes. If people can pop in, Moira will take care of that. But yeah, let's take a few more questions now.
Okay, while we're going through the questions, I'm just going to push through a poll to everyone. So this is going to go live, and then we'll carry through with the questions. All right, so next question we have is, What is the shelf life for reagents, assuming all are kept in a controlled temperature environment?
I believe that should be captured.
It does depend on each individual reagent, and it will say on the bottles. For example, I'm looking at the reagents that are the enzyme here. So this is stable for 3 months after hydration if you store it in a refrigerator, so 4 degrees centigrade. Stable for 6 months after hydration if you freeze it to -20. And otherwise, it has an overall expiry date based on the lot number.
So that will be captured.
Sorry, Virginia, to interrupt. I should say it's over a year. This particular one was resuspended about a month ago. It's been sitting in our 4-degree fridge, and it doesn't expire until July of 2021.
Mm-hmm. And we will have all that information available online or in the handbook as well. And again, if you have any questions, let us know. But we do want to understand that a lot of these applications are buying kits and running them perhaps once every week or maybe a couple weeks. So they will last a long time. We want to make sure that they are lasting on your shelf as well. Again, avoid the high temperatures. Keep in a dark place. The fridge is always the best place.
This question for the most part was answered, but just to confirm or reaffirm, the average life expectancy of the enzyme once you activate it with the buffer.
Yeah, so once I activate it with the buffer, again, it's all going to depend on how often you use it, but if you're treating it nice, it's in the fridge right after testing and doesn't get heat shocked at any moment. And by heat shocked, I mean, again, 30 or 40 degrees centigrade for a period of time. You could expect this to last for months.
And anybody who's been with us for a long time will know that those are now in smaller bottles, and that's for the same reason, so that you don't have to hydrate the full 100 tests in your kit. You can hydrate a small amount and use that, and everything else will last longer until you're ready for it.
And again, just because we run that standard ATP test, the SRLU value, you don't need to guess whether or not your enzyme is healthy or not. It's a yes or no based on the results of that positive control.
If you're doing daily measurements, how often do you have to do your controls? Is it just one time per day, or is there regular intervals at which you would do a control?
I definitely would be doing my controls every day. If I am taking multiple samples throughout a day, again, I like to think of this as in batches, and I'm going to be running those controls for each batch. I wouldn't rely on a standard or a positive or negative control taken at 9 o'clock in the morning when I'm testing at 4 o'clock in the afternoon.
This one might be a bigger conversation, but by what conversion factor are you relating ATP to microbial equivalents?
Yep. So this is very loosely laid out in the handbook, but I can tell you that that conversion factor is based on a pure culture of E. coli. So there are a number of assumptions there, including the load of ATP per cell volume. That has been worked out, and I have done or scoured the literature for kind of an average load of ATP per cellular unit for environmental isolates, and we're able to use that number to, again, convert the mass of ATP back to a cellular load. It's not going to be the most perfect method in that there is some slight variation, but once more, that variation that you see isn't significant enough to affect the log scale based on the study that I conducted.
And again, if you'd like to just use the picograms per mil of ATP units, that's also completely valid.
Can the ATP methodologies shown today be used within municipal wastewater or industrial water treatment?
100%.
Okay. Within a water treatment scenario, have you guys come across any additional interferences beyond what you've demonstrated today that interfere with the enzyme?
Yeah, so the big one that I have seen out there is with oxidizing biocides, so things like chlorine dioxide and bleach. If you attempt to treat a water with your oxidizer and immediately test it, and I mean immediately, as in within minutes afterwards, the presence of that oxidizer within that water will disrupt the activity of the enzyme, because once again, that enzyme or protein is a living thing of sorts, and it will denature from the addition of that type of biocide. If you can wait a couple hours, even 30 minutes starts to show some reduction in that. If you have any concerns around biocides and stuff, I do have a couple slides I can share with people that showcase what is affected, or more importantly, what is not affected. Because in most cases, the addition of biocides isn't that detrimental at all to the measurement of ATP.
And the nice thing about oxidizers is that they react quickly and they're gone. So again, it typically happens only when you're dosing at really high concentrations, which in most of our applications we don't. So if you're dosing at demand and you give it enough time that it gets completely consumed, there should be no interferences. But again, depending on how you're conducting this type of testing, what you're trying to get out of it, we can provide some guidance on that.
Yeah, the last thing I want to add to that too is, again, think about the steps within that planktonic testing that you're going to be looking at that water. You're doing a filtration step to get just the microbial cells and any other contaminants or debris, let's say, on that filter membrane. So a lot of your chemical is already going to be washed away through that filtration step. And then if you're doing a rinse step on top of that, really that is quite an effective way to deal with these concerns.
Can you use sterile filtered water to rinse the sample instead of using a rinse reagent?
I would not advocate that. There are some other things in the rinse reagent outside of water that are going to be important in that step. Proprietary, thanks.
Is there a significant difference between species and their total cell ATP?
Yeah, we've been talking about this a few times already. Different species will have a different level of ATP per cell. But again, the data that comes out of this test at the scale of a log unit is going to be highly accurate in this, or it'll meet your needs for testing control programs and understanding active microbial loads.
Is filtration required for, like you've shown earlier, a sludge style sample? Will the solids in the sludge interfere with the RLUs generated and measured?
So filtration is always going to be the best way to go. If you are doing that non-filtration method, adding it directly to this larger tube, yeah, it is possible that things you are adding in there are going to slow down or inhibit some of the action of the lysis agent and of the assay in the end there. And that's why I said that, one, this is a workaround for tricky samples with high sludge, and also for all samples that you want to compare, making sure that you're sticking with this method. Because then at least all of your samples have been exposed to those same detriments of sludge and other things that might be inhibiting it.
And we provide these workarounds because we've gone through a number of iterations of trying to find the best way to deal with these challenging samples. It's the nature of our industry.
Yeah.
And the ones that we are presenting today have been the best ones to get the best results out of it. Microbes are delicate. If you put too much work behind it in order to try to pull them out of that sludge, we end up damaging them, or you lose that, or it's an inaccurate reading at the end, or closer to being inaccurate.
With regards to surface swab and solids testing, what sample preparation is recommended?
So surface swab testing, sample preparation, I'm going to say as little as possible. In this example here, we had a pipeline cutout. Somebody had to take some sort of cutting tool, a grinder, and actually section this pipe. So there's going to be heat involved with that, handling involved with the sample. You definitely want to try to protect the surface that you are sampling from any outside forces as much as possible. The other thing that I'll see quite often is somebody will pass in a sample and say, oh, I sandblasted it for you. It's ready to go for microbial testing. Yeah, that's a big no-no, of course, because that sandblasting is abrasive and it's going to remove that biofilm community there. Even just so much as, you know, avoiding that human nature of wanting to touch and poke at the cool feature that's on there. If you're not wearing gloves, or even if you are, there could be other things on your fingers. And then when we do DNA testing, believe it or not, we can pick up the traces of those human skin microbes from, you know, all the busybodies.
There's no hiding with the DNA testing.
So yeah, handle that sample as little as possible.
Can the photon master be connected to a computer to allow for data processing directly from the instrument itself?
Yeah, so we only use 2 buttons on this instrument. It can be hooked up directly to a computer. OSP does not currently have software directly that you can use for that. There is some other software opportunities out there to do that. But in all honesty, I find just writing that number down here and plugging it into the calculator spreadsheet that already mimics and matches with the testing kits that we're pairing it up with tends to be the most robust, least confusing way to do it.
We like to allow as much adaptability within what we're doing. So again, we may add procedures or adjust our calculations to accommodate our client's needs. For that reason, it's easier to manipulate a worksheet rather than going directly into some software. Again, doesn't mean that it's not accessible. Please let us know, but there is options.
How long after sampling the water source can you store the sample prior to running the ATP test?
Ooh, good question. We actually didn't talk about that a ton. So you want to maintain a sample in a way that the microbial community doesn't change from the point at which it was sampled. As in, if I take a water sample and I ship it and it takes a week to get to the lab and it's experienced all sorts of different temperature and pressure conditions and you name it, it's highly possible, as in I'll say it's going to change the microbial community. So as much as possible, best practice will dictate that you do this testing at the source and as soon as possible. If that's not an option in your situation, that doesn't mean that you can't process a sample. It just means you have to appreciate that for the amount of time that the sample hasn't been processed, if it was frozen, if it was heated, so on and so forth. All of these things will change the active microbial load in your sample, and you just have to accept those risks.
We are down to our final 4 questions. I appreciate we're going over time, but we'll roll through these last 4. Can you use ATP testing in finished fuels?
Absolutely. We talked a lot about the contamination levels for oil and gas samples. The only difference between this process and finished fuels is that you're going to have different contamination levels. Normal fuel, standard fuel, no problem, exactly what we went through. If you are testing aviation fluid, they tend to have contamination levels that are much lower, and we would simply provide a different enzyme in that case. But again, we have those kits available.
Can you comment on the use of ATP to evaluate surface active biocides such as a glute?
Sessile testing.
Yeah, sessile testing after an application of the biocide, and then comparing your before and after biocide treatment.
Can you use ATP testing to track biocide treatment efficacy or just for evaluating nominal bacteria levels?
We do kill studies all the time. Any type of biocide selection study, we recommend that you use the ATP test kits. Part of the reason is because our biocides are not selective. They will kill everything equally, which means that the amount that they're going to have to work and the amount of control that they have is going to be based on your entire microbial load. So using the ATP test kit to get a total microbial count before and after is the best test method that we would recommend. But absolutely, we can use it to evaluate any biocide application.
Is there a large difference in ATP results between a sample collected in the field and then read in the field compared to a sample that is shipped on ice, let's say, and analyzed 2 to 3 days later?
Yeah, there certainly can be. I won't go too far into this because we did talk about it a little bit before, but yeah, by the time you ship a sample, the microbial community could change. So again, best practice would be to do it in the field. And if you can't and you have to ship it in, just make sure you're staying consistent again amongst the samples that you do want to test and compare.
And keep it cool, don't freeze it. That's another key thing.
Freezing kills.
Okay, we had 2 more sneak in, and then so this will be, and if there's further questions, please feel free to connect with us after. But just to go to these last 2, does the handbook include suggested kill study methodology?
No, the handbook is specifically for the ATP process. We have no problem giving you some guidance on how to create certain kill studies. They will be different for different applications, but by all means, reach out. We can help you through that.
And hopefully I don't botch this question. Bear with me. Can you store this sample between 40 to 42 degrees Fahrenheit for 8 hours and still preserve that bacterial load?
What's 40 degrees Fahrenheit?
Come on, guys, we're a mess. No. Again, as long as it's below, if you're around a cooler temperature, not freezing, that's the temperature. And yes, you can store for 8 hours. It doesn't mean that anything is not going to change in that sample. we try to limit growth more than anything when we lower those temperatures. But just understand that, yeah, there's going to be limitations based on how far out you go with that.
Yeah, it's not a line in the sand to say that what you've done means the sample can be tested. It's just you do the best that you can in the conditions that you have to try to prevent microbial changes.
Okay, so that's all the questions we have.
Excellent. Thank you so much, everybody. We really appreciate it. Let us know if this style of webinar was good. We are available to do more. Give us some suggestions. Otherwise, thanks again. Have a great day. Have a safe day. We'll talk to you soon.
Thanks, everyone.